Hello everyone. In this session, we will be looking at how to draw the stratified squamous keratinized epithelium. And also we will see a few common mistakes committed while drawing these diagrams. This is a diagram of stratified squamous non-keratinized epithelium. In part 1 of the session, you have seen how to draw this diagram. If you haven't watched the first part, check the link that is given in the description. Once you know how to draw this diagram, in order to make it a keratinized epithelium, all that you have to draw is like this. Adding a layer of keratin superficial to the squamous cell makes it into a diagram of stratified squamous keratinized epithelium. This is how a complete label diagram will look like. What are the common sites in our body where these two types of epithelia are seen? The part of our body that is most exposed to the outside world is skin. So in our skin, we want an epithelium that offers the best protection. For that, which epithelium would you prefer? Do you prefer a single layered or a multi-layered epithelium for offering a better protection? We definitely need a multi-layered epithelium with as many layers as possible. If you look at the different epithelia across, you can see that from the simple to the various type of stratified epithelia, it is the stratified squamous keratinized epithelium which has the most number of layers, 8 to 10 layers. Even though it is called as stratified squamous, it is not the squamous cells which are present in all the layers. You have the polygonal cells, the basal layer of columnar cells, and finally only in the topmost layer you have the squamous cells. So it is this epithelia, the stratified squamous keratinized epithelia, that we want in our skin with this extra layer of keratin on top of it. So the sites where you see the stratified squamous keratinized epithelia are thin skin and thick skin. Thin skin that is present throughout our body, and thick skin is the skin of the palm and soles. So in both these areas you have stratified squamous keratinized epithelium. What about the common sites of non-keratinized epithelium? Just like its counterpart, this type of epithelium is also seen in areas which are exposed to the outside world. There is an add-on. The non-keratinized epithelium is seen in moist areas which are exposed to the outside world. If you take your face, what are the moist areas on your face which are exposed to the outside world? One is the oral cavity, which is a major structure present in the oral cavity. It is the tongue. So the epithelium of tongue is stratified squamous non-keratinized epithelium. Similarly, the tonsil, which is present in the oropharynx, also has stratified squamous non-keratinized epithelium. Once the food enters the oral cavity after chewing, you swallow it, and through which region does it reach the stomach? It is through the esophagus. So esophagus is immediately in contact with the outside world just after the oral cavity. So what will be the lining epithelium of esophagus? It will also be non-keratinized epithelium. So the sites of stratified squamous non-keratinized epithelium are tongue, tonsil, esophagus, and can you think of any other structure in the face which is moist and is exposed to the outside world? Obviously the eye. So in eye, the cornea is again having stratified squamous non-keratinized epithelium. If you look at the lower part of your body, what are the moist areas exposed to the outside world or immediately in contact with it? the distal part of the anal canal, the distal part of the vagina in females, the distal part of the urethra, all of them are having stratified squamous epithelium. So these are the common sites for these two types of epithelium. Let's see some of the common mistakes while drawing these diagrams. This is a circle inside which you draw the diagram. This represents the field of the microscope through which we view the specimen. There are certain specimens which fill the entire field, so the diagram will be filling the entire circle. Then there are other specimens which are more linear, like the epithelium, which occupy only a part of the field, not the entire field. So we draw this diagram only in a certain part of the circle. So where do you want your diagram to be? Is it in the upper part, lower part, middle part? So what is aesthetically more pleasing is if you draw the diagram in the middle 1/3 of the circle. Now this area, the middle 1/3, is where you want your diagram to be. The mistake number 1 that the students make is to start drawing the basement membrane from the center of the circle. So what happens here? So this will be the diagram at the end. So you will have the entire diagram shifted up and you will not have enough space above. So you want to bring this basement membrane a little more down so that the superficial layers can also be drawn comfortably. Here an imaginary line is drawn through the center of the diagram. Here you can see the relation of the basement membrane with respect to this equator. It is drawn, the basement membrane is drawn a little below this central line, so that when we complete the diagram, the entire diagram will occupy the central part of the field. Another mistake that happens is while drawing the papilla, that is these dips. So here the basement membrane are too close to each other. The bends of the basement membrane are too close to each other. Now what is the problem with this? The problem is that when you draw the columnar cell, there will not be enough space to draw the columnar cells of these two areas. There will be a lot of crowding of the cells. So while drawing the basement membrane, maintain a little gap in these dips, this papilla, so that you have enough space to draw the columnar cells of these regions. Once you have drawn the basement membrane correctly in the correct location, the next step is to draw the simple columnar cells in the basal layer. To see how to draw the simple columnar cells, please watch our videos regarding how to draw the simple columnar cells. Now specific to this diagram, what can happen is like this. When the student start to draw the columnar cells, they will be of a uniform height in this region, whereas when the dips or the papilla in this region, there is a tendency for many students to draw the columnar cells as very tall. So if you compare the height of the columnar cells throughout, if you compare these two areas, there is a wide discrepancy in the height. So this can be avoided. How? This is a reference line which is parallel to the basement membrane, which shows that the height is quite different. So initially, before drawing the simple columnar cells, draw a faint reference line like this and then draw the columnar cells so that all of them are of uniform height. So once you draw a basement membrane, the next step is to draw this faint reference line parallel to the basement membrane just above it. This reference line is not a part of the specimen. It is a guide for you which will help you to draw the columnar cells with uniform height throughout the specimen. Mistake number 4, blowing it out of proportion. Here the shape of the cells are correct, but the comparative sizes of the two cell layers, they do not match. Here the polygonal cells are drawn way too big when compared to the columnar cells. The size of the polygonal cells should have been a lot more smaller and it would have been correct. So how do you know what is the size of each cell layer? For that, there is no other option other than to look into the microscope and see the size of the cells yourself. You have to compare the different cell layers and see the proportion of the size of the different cell layers. And then only you can draw the cell layers matching with one another, so that the proportion of the size in each cell layer go together as one unit. So here the shape is correct, but the proportion is wrong. Finally, drawing a straight basement membrane for a stratified squamous epithelium. Is it a mistake? Instead of having the papilla, instead of having those dips, drawing a straight basement membrane so that the entire epithelium looks like this. It is not a mistake, because in cornea, this will be the appearance that you get. Cornea, which is a site for stratified squamous non-keratinized epithelium, the basement membrane does not have any papillary appearance. It will be a straight line. So is it a mistake? Well, it is not. But in majority of the sites, the basement membrane have that papillary appearance. Thanks for watching. If you find our videos useful, like, share and subscribe Club Anatomy.